Hello and welcome to this week's Health Report with me, Norman Swan. Today, how we really are what we eat, how our food changes our inner health, as the ads like to say, and the secret ingredient of the Mediterranean diet. Also, making medical research more productive and important Australian research just published in a major international journal. The findings from the Centenary Institute and Concord Hospital in Sydney could make transplantation easier, enable hepatitis C to be treated more effectively, and maybe also autoimmune diseases like insulin-dependent diabetes and rheumatoid arthritis. Professor Jeff McCaughan is Assistant Director of the Centenary Institute and Head of their Liver Lab, and Patrick Bertolino is a Senior Scientist at the Centenary and one of the main people driving the work.
Patients that receive an organ transplant, they need to receive immunosuppressive drugs, otherwise this transplant will be rejected. What we know is that the liver seems to be different, and if you transplant the liver together with the kidney and heart in animal models, these kidney and heart that are normally rejected are now accepted because of the liver graft.
Really?
Yeah, so it's quite an amazing property that we have known since the 1970s, and it has remained a mystery for all this time. So we think that we have basically discovered a process that might explain this property of the liver.
And you made a discovery a few years ago that's led to this one now.
Yes. So what we found 10 years ago was when T cells, which are, so there are different types of T cells, but some are killer cells, so they will be able to reject a transplant.
And just so the audience just follows us here,
Yes.
and actually they can listen to last week's health report and find out what T cells do, because we had Jack Miller on who discovered the T cell.
Okay, yes.
But essentially these are the attacking white blood cells that are involved in fighting viral infections, but also when you transplant an organ, they're the ones who reject the organ.
That's correct, yes. So we have observed in mice that when these T cells go in the liver, they seem to disappear. So we found them in the beginning and then they disappear.
So it's a black hole for T cells?
It's a black hole, exactly.
And they didn't reject the liver?
And they didn't reject the liver.
So these white blood cells, T cells, which were getting ready to attack foreign tissue, were consumed by the liver, never to reappear. Like a haunted house for the immune system. Jeff McCaughan.
In the liver transplant situation and potentially in viral infections, exactly. This is an effect which seems specific for the liver.
So you've suspected this for a while?
Yeah, we suspected this for a while, but we didn't understand it. We didn't have all the pieces of the puzzle, if you want. And in 2004, a collaborator working at the Concord Hospital, Alison Warren, showed me some electron microscopic pictures and showing that basically some of these cells were going inside the liver cells, which are called hepatocytes.
So he had the smoking gun. He actually had a photograph of the crime in action.
That's correct. That's correct.
Well, it wasn't a crime in this case.
So this was really puzzling. And actually this process has been described in the 20s, the process by which a cell can go inside another cell. But what we've discovered down the track was that these cells, once they enter the hepatocytes, they get destroyed.
Hepatocytes being liver cells.
These are the liver cells. So they were destroyed, and probably that's why we couldn't find them. So this was the real discovery, that basically these T cells go inside hepatocytes, these liver cells, and then get eaten up.
And the movie's fantastic because the cell, they just don't get destroyed. They sort of go inside the cell, and Patrick's got beautiful movies where they wander around the cell.
Wondering where the hell they are.
Wonder where the hell they are and what are they doing in such a weird environment, and then they get destroyed.
So Jeff, I mean, your expertise is in the liver. I mean, the liver is your organ.
Yes.
What did we think the hepatocyte did, the liver cell did, before this discovery came along?
Well, we certainly didn't think the hepatocyte did this or had any role in
the
this
immune
type
system.
of function.
So are liver transplants more easily accepted than heart transplants?
Oh, yes, much more so.
So you use fewer anti-rejection drugs on people who received a liver transplant than a heart transplant?
Exactly. But what's really intriguing about the processes that Patrick's described is this happens very, very early. This happens within hours of a transplant. And this is also a time where we're giving maximum anti-rejection drugs. So one of our queries is whether our anti-rejection drugs, which we give early to stop rejection, potentially interfere with this wonderful natural process which could actually allow the liver to adapt with much less immunosuppression than we even give now.
So you're thinking you're dealing with a heart here, but in fact you're dealing with a very different situation.
Exactly.
But just go back to my original question. I mean, I know the liver as a detoxifying organ. So the liver does a few things. It produces cholesterol, it manufactures substances, clotting substances for your blood. There's all sorts of things the liver does, but it also detoxifies.
Correct.
And is that what the liver cell does, all those different functions?
Yes, and this is an immune cell interacting with a non-immune cell. This is why it's so fascinating.
So let's just summarise where we got to, because it is complicated. What you're finding is that when you've got T cells that have been fired up by the immune system, they're angry, they want to get this foreign tissue, and it's the liver that's activating them, am I right here? Yes. If you like, the sniffer dog has been sniffed against the liver, and they come into the liver has a mechanism in these cells for chomping up, swallowing up and chomping up these aggressive anti-liver T cells.
That's correct. But it does this at a time when the cells are not yet killers.
So they're still in nursery school, they're still in playgroup.
That's right.
Absolutely.
Now, could this be a bad situation? We sometimes need these T cells to mop up cancers that might have arisen or viral infections. There's all sorts of things these T cells do that we need. Could it be that we get rogue liver cells that chomp these up when we actually need them, Jeff?
Well, this is the classical situation in hepatitis C. Your nursery cell, if you like, against hepatitis C will first see hepatitis C in the body on a liver cell. And this phenomenon, as Patrick's shown, takes out the most effective killer T cells. The highest potent killer cells could be taken out by the hepatitis C virus at exactly the time that you want to activate them.
So what's good in transplantation is bad in hepatitis?
Absolutely.
Yes, that's what we think. Yes.
In all forms of hepatitis or just hepatitis C?
Well, one of the fascinating things is that hepatitis C, when you get infected with hepatitis C, most adults never clear it, while hepatitis A, which comes through the gut, not from the blood, is always cleared. So our hypothesis is that if you see the virus for the first time in the liver, then these killer cells will be got rid of and the virus will stay, while in hepatitis A perhaps...
Because you saw it in the food and therefore the immune system
has already
sorts it out
seen
in the food.
Yes. Mm-hmm.
So where to from here? You've already said, Jeff, that this might mean that you could do a trial of cutting back a bit on the anti-rejection drugs straight off to see whether or not there's a more natural process here.
This reinforces that perhaps we should really do that. And the other thing about the anti-rejection drugs, of course, is that long-term, these drugs, even in small quantities, predispose to cancer and to cardiovascular disease. So the concept that we could use this process to even withdraw patients from immunosuppressive drugs after several years is very, very enticing and intriguing.
And in hepatitis C, is there a therapeutic angle here for hepatitis?
Well, Patrick's investigating inhibitors of this process, and that might improve outcomes for hepatitis C.
You want to play both sides of the fence here,
yes,
Patrick?
This opens up a lot of possibilities. We could develop a new class of drugs that prevent or promote this process, and we can change the outcome depending on whether we are looking at transplantation or hepatitis C treatment.
There was just one little anecdote. I presented this work in front of a group of well-known liver pathologists internationally, and one of the senior liver pathologists who's been around for 30 or 40 years, he said, "This is just amazing, Jeff. It's fantastic." He said, "We've known about this." I went, "What?" And in the 1970s...
So these geeks have been sitting with...
So these geeks used to do, it's gone out of fashion, electron microscopy on human liver biopsies. And he said, we used to see this all the time in viral hepatitis and in autoimmune diseases.
They just didn't think to tell anybody?
They just didn't. He said, go back to the textbooks and you'll find it, and you can find it. He said, we just didn't know what it meant. We thought it didn't mean anything. So this process is happening in humans.
And another use could be to engineer liver cells to put on a coat which makes them look like arthritic joints or clapped out insulin producing cells, and thereby attracting the T cells involved in, say, diabetes or rheumatoid arthritis, then gobble them up. Jeff McCon and Patrick Bertolino are both at the Centenary Institute in Sydney. And this is the Health Report here on ABC Radio National with me, Norman Swan.